Hello, fellow researchers. We are Mela, Marcus, Jan, and Ivan from the Institute of Computational Biology at Helmholtz Zentrum Munich and the Technical University of Munich. We are glad to present you our work on extending mvTCR with pre-trained T-cell receptor large language models. Advances in single-cell sequencing enable the simultaneous measurement of the transcriptome and T-cell receptor, in short, TCR sequences, at cellular resolution, and promise to offer new insights on adaptive T-cell immune response. However, it is still an open question how to integrate these two modalities. The multimodal generative model mvTCR was developed to solve this problem. It tries to find a joint representation of TCR sequences and single-cell transcriptomic data. This helps to understand T-cell responses by identifying groups of cells with shared phenotypic and functional profiles reacting to the same antigen. Additionally, in recent years, several large language models have been developed to improve TCR sequence representation, profiting from self-supervised representation learning on large unlabeled datasets. In our work, we combine these advances by replacing the sequential input and transformer-based architecture of mvTCR with embeddings of TCRs from different large language models.
We benchmarked our models on two different datasets, 10x Genomics and Fisher. The 10x Genomics dataset contains single-cell RNA-seq data, TCR sequences, and in vitro measured antigen binding specificities for CD8 plus T cells. We compared the models with respect to two features that represent different aspects of the dataset: cell type for cellular heterogeneity and antigen specificity for functionality, each analyzed by the F1 and the NMI score. Every mvTCR model with a large language model embedding as input shows at least a tendency for a better conservation of the cell type in comparison to the vanilla model. For antigen specificity, the large language models achieved similar results to the vanilla models, where the deep TCR and TCR bird models show a trend for a better conservation. Our models achieved similar results for the Fisher dataset, containing single-cell RNA-seq and TCR sequences of 2 SARS-CoV-2 reactive patients. Here, reactivity is reflecting cellular functionality in the context of COVID. Most models using large language model embeddings have at least a tendency to capture the cell type better while preserving reactivity. Among those, deep TCR trained on the large TCR sequence database, TCR-DB, performs best.
For a further visual comparison, we created UMaps of representations learned by mvTCR for each tested model and different aspects of the 10x Genomics datasets: clonotype, cell type, and epitope specificity. On the first look, TCR bird is different with large overlapping clouds, similar to the RNA-only model, whereas the other models form smaller, more concise clusters. Taking a look at the binding specificity plots, most epitopes are well separated on a local scale, but are distributed over multiple clusters for all models. The coloring by clonotypes is in accordance with these small subclusters, indicating that most clusters correspond to individual clonotypes. The cell types are shown as a gradient in all models. Overall, the UMaps show that the models using deep TCR and T-Rex embeddings capture those features best.
To conclude, we showed that mvTCR benefits from using large language model embeddings. Among all tested models, the one using deep TCR trained on the large TCR-DB database performed especially well. This work further contributes to learning multimodal representations of single-cell immunoprofiling data for integration, clustering, and specificity analysis.